Welcome to lecture of mine on the topic WHO Guidelines for Quality Control and the Standardization of Herbal Drugs. This has been provided by Department of Pharmacy, SRMCT Bareilly. The contents that we'll be discussing today are in the particular manner. Firstly, we'll be reviewing some of the important definitions, then need and safety of quality control and management. Further, we'll be seeing into the constraints and the three pillars of ideal herbal drug and the rational use. Also, we'll be discussing the points given by WHO regarding the standardization of herbal drugs or crude plant parts. Finally, the flow diagram and their analytical procedures. Now, a very important thought. Herbal medicines are the oldest form of healthcare known to humanity and has been used in all cultures throughout the history. Now these are the few definitions. We'll be looking one by one. Firstly, what is quality? Now this is the status of a drug which is determined by identity, purity, content and various other properties. The safety of the drug assures that it does not have any side effect, whereas purity assures that the drug is not contaminated with potentially harmful substances. Further, potency can be described as the content of active constituents within the defined limits. Now, various identical process refers to the involvement of standardization in maintaining the quality and validity of the manufactured product. Now, what are herbal preparations? These are obtained by scientific treatments such as extraction, distillation, fractionation, etc. These include comminuted or powdered herbal substances and various tinctures, extract, essential oils, and process exudates. Whereas herbal substances can be defined as the various plant parts used and not being subjected to a specific treatment, which are also considered to be under this following category. Now, markers are chemically defined constituents or groups of constituents of a herbal substance which are used for control purposes. Now, the need of quality control. The quality control and standardization process helps in the reduction of adulteration with substandard drugs. Also, problems arising due to improper authentication can be sorted out. There is scientific validation of the therapeutic claims. Identity, purity, efficacy, and safety of the drugs is maintained. And customer satisfaction and confidence also arrives. Now, what are the various problems while considering the safety factor in quality management? They can be summarized as adulteration with other medicines, mistaken use of wrong species, incorrect dosing, misuse of herbal drugs, various herb-drug interactions, toxicity parameters, poor or substandard quality products, and a misconception that all herbs are safe. Now these are the constraints in quality control of herbal drug products, starting with regulatory aspects, then adulteration or substitution of substandard products, no certification of raw material, that is a major issue. Then, no data of pharmacovigilance. Also, there is a lack of trained manpower and poor investment in R&D of herbal manufacturing of products. There is a lack of public test houses and safety evaluation facilities. So these problems are various, or you can say a huge, in terms of main dependence on the file sources. Now these are the three pillars. The first one is quality, second one is efficacy, and third one is safety, which are very ideal for the herbal drug and their rational use. So for appropriate use of herbal drug, we have to consider the quality, safety, efficacy, accessibility, and availability. The World Organization is a specialized agency of the UN that is concerned with the international public health. It was established on 7th April 1948 with its headquarters in Geneva, Switzerland. Now, it provides various guidelines for prevention, control, safety, efficacy, as well as evaluation and standardization of herbal materials. For example, these are the basics or guidelines laid down by WHO in 2005, 2002 A and subsection B, similarly in 99 A and subsection B, likewise in 1998 A, B, 1996 A and subsection B, 1990, 81 and 79. These are the WHO guidelines and their front covers which have been given all through these years. Now, a set of guidelines for the assessment of herbal remedies has been published by WHO. These guidelines specify criteria to be used in evaluating commercial crude drugs intended for use. Also, the guidelines on safety monitoring of herbal medicines in pharmacovigilance system has been laid down in 2004. Similarly, a QC for medicinal plant was given in 1998 and for GMP in 2005. These are the various WHO monograph on selected medicinal plant covering under the various volumes of 99, 2004, 2007 and 2009. Guidelines have been given for assessing quality of herbal medicine with reference to contaminant and residue in 2007. Also, few basic tests for drugs, their pharmaceutical substances, medicinal plant materials, and dosage form are laid down in 1998. In 2000, guidelines have been laid down for methodologies on research and evaluation of traditional medicine. Similarly, in 2003, a GSEP for medicinal plant, that is good agricultural and collection practices, has been given. In 1993, guidelines has been given for evaluating the safety and efficacy of herbal medicines. In 1998, regulatory situation of herbal medicines, a worldwide review has been given. Now, what is standardization of herbal drugs or crude drug plant material? It covers various categories, starting with definition, which includes name of the plant, part of the plant, and in which condition it has been collected, that is whole, powdered, fresh, or dried. Then go for authentication or confirmation, that is correct geographical origin or correct stage of growth is necessary. Third is absence of foreign material. It means the foreign matter or material should not be more than 2%. Now, this foreign material or matter can be described as the other plant part, which comprises of soil, stones or dust, or insects or some other animal material. Then, microscopic characteristics which leads to the confirmation of the identity can be described in qualitative features or quantitative features, that is terminal number, vein islet or termination number, palisade ratio, etc. The radioactive contamination limits which arises from environmental pollution or microbial decontamination process. For this, a assay for material containing constituents of known therapeutic activity or known marker compounds. Non-specific assay methods for groups of compound may be used where specific assay methods are not available for single compound. Now, the processed plant material or non-plant material, that is extract, tincture or combination. Now, what are the various guidelines given by WHO to standardize these combinations or tinctures? The first comprises of the definition, that is under what category it comprises, that is whether it's a liquid, a solid or etc. Then you have to look after the organoleptic characters like macroscopy, smell, taste, texture, color, odor, etc. A chromatographic profile can be used to confirm the presence of unique compound, to confirm the TLC chromatogram, to confirm the HPTLC chromatogram, Also for hygroscopic material, we can consider the water content or moisture content here. Now various ash values like acid insoluble, total ash, sulfated ash can be determined. This ash value is a major criterion which is used to indicate the extent of contamination with inorganic material and is determined by the incineration process. Now the presence of volatile material for specific plants which contain volatile oils can be determined by the steam or distillation process. Now the powder material can be looked out by the test method and acceptable limits for particle size or distribution. If the product is a mixture of plant material, the supplier must provide evidence that each component has been individually tested for heavy metal limits, microbial contamination limits, as we have discussed in the previous slides. Also, the pesticide residual limits which arises from the cultivation has to be found out. The extractive values for different solvent indicates the proportion of polar and non-polar components. Now this is a flow diagram for the quality evaluation of herbal drugs, which can be carried out under several categories like organoleptic, botanical, physical, chemical, and biological. Here you can see organoleptic comprises of all the measures, all the identical tests which can be done with the help of sensory organs. For botanical, these include two types of tests: macroscopic, which are done for the shape, external markings, and microscopic for qualitative, quantitative, or powder studies. The physical criteria comprises of the calculation of moisture content, extractive values, ash values, and analysis of the fluorescence. The chemical comprises of qualitative and quantitative tests, which can be done by various HPTLC, GLC, HPLC, or any of the chromatographic methods. Under biological evaluation, it can undergo for the testing of the microbial contamination or some toxicological or pharmacological specific activities. For analytical procedures, the botanical parameters which can be considered are sensory, foreign matter, and microscopy. The sensory evaluation means which includes visual microscopy or touch or odor or taste. The foreign matter including foreign plants, foreign animals, or foreign minerals, etc. The microscopy can be done by the use of razor blades, microtome, which includes histological observation and measurements. For physicochemical methods, ash values like total, acid insoluble, water soluble, acid soluble can be calculated out. Extractive values can be calculated in hot water, cold water, ethanol. Also, ether soluble extractive values can be found out. Loss and drying can be done in case of volatile matter. If any volatile oil is present, can be estimated by the process of steam distillation. Further, there can be determination of crude fiber content or moisture content. Certain bitterness value, hemolytic property, astringent property, swelling index, and foaming index can be done to confirm the biological parameters. So if we consider the parameter of bitterness value, this is the unit equivalent to the standard solution of the quinine hydrochloride. This hemolytic property is done on ox blood by comparison with the standard reference solution of saponin containing drugs. The astringent property, basically the criteria of the tannins which bind to the standard Freiburg hide powder. And if a drug constitutes some mucilage or gums, now they will show swelling index when they are swelled in water, or foaming index in case of saponins are present, that is foam height produced by 1 gram material under the specified conditions. For toxicological parameters, these include the various identification procedures. In case of arsenic, a stain produced on HGBr2 paper in comparison to the standard stain. For pesticidal residue, which includes the total organic chloride and total organic phosphorus. Various heavy metals can be identified. Similarly, aflatoxins presence, radioactive contamination presence, or heavy metal presence can be found out to give a safe limit in the toxicological parameters. Now these are the references for your kind concern which you can use to understand what we have just learned, that in very short what is quality control and what is standardization procedure. If you have any problem, kindly go for the references further. So thank you very much for your patientful listening.